Okay, you are. Great. Okay, thank you everyone for being here. I'm very happy to introduce Charlotte Craig. Before joining us in 2021, Charlotte completed her undergraduate degree at the University of Toronto working with Dr. Jane Malcolm in forestry. There she had lots of experience working in remote and buggy and sweltering summer conditions in Algonquin. And this signaled to me that she'd be able to thrive easily in these trying field conditions I'd been cooking up for her project. That asked, what happens to pollinators when we make summer, even hotter? Charlotte perseveres, she's tenacious, and she sees the end goal all through her master's. Using some sports lingo, she did play basketball through undergraduate, so she's been keeping her eye on the prize all through her master's. She's never wavered through multiple methodological trials and tribulations, theft of material, equipment malfunctioning, and of course, very, very hot summer temperatures, as you'll see. She's been an easygoing, responsible, motivated, and overall very fun person to work with since day one. And she's trained multiple undergraduate students, made great friends in the lab, some of whom are here today. And she's maintained her relationships with important partners like campus facilities, the UTSC farm, and our horticultural growers for lots of our projects. And so I'm really happy and excited for all of you here today to witness Charlotte's presentation. And so Charlotte, please, thanks again, and take it away.
Thank you so much. I was so kind. Alrighty. So again, thank you all for coming. I'm excited to present my master's thesis, which is entitled Experimental Warming of Flower Temperatures and Influence on Pollinator Visitation. So let's get into it. So the synergistic effects of climate change in the urban heat island are causing Toronto to warm at an alarming rate, with average temperatures and number of extreme heat events steadily increasing. Toronto already experiences the highest number of extreme heat events in the country, with this frequency predicted to triple between 2005 and 2050. These increases in temperatures cause humans and animals alike to be at risk. One group that is at risk is plant-pollinator interactions. So increases in temperature in cities has the potential to alter plant-pollinator interactions because both pollinators and plants heavily rely on temperature to effectively function. Ectotherms, such as bees and other pollinators, depend on external sources of heat to regulate their bodily functions. And as such, they compensate through behavioral changes to maintain their thermal optimum in their environment. Some bees shiver by contracting their flight muscles to stay warm on cool days, while other bees take cooling flights to stay cool when it gets too hot, as well as many other interesting behaviors I'd love to talk about after. Bees have so many different behavioral approaches to make because they have many different behavioral changes to temperature fluctuations because they're an extremely diverse group and they have different temperature limits and requirements. There's many factors that impact temperature preference and climate sensitivity, including taxonomic differences, as well as ecological traits such as sociality, body size, or specialized foraging behavior. For example, some studies have shown that social bees are more heat tolerant than solitary bees, suggesting that life history may predict climate sensitivity. And studies also show that smaller bees are definitely impacted by changes in temperatures than their larger counterparts. Additionally, the time in which bees forage can change their ability to stay within their thermal range. Like some bees that forage in the hottest peak of summer, they are threatened most by the increases in temperature and extreme heat events when compared to bees that forage in the spring and late summer. Well, there are many different factors that impact the thermal sensitivity of bees. The critical thermal maximum, or CT max, and the thermal tolerance range have been identified as the two most important. CT max is the maximum temperature that an individual can survive, while the thermal range is the tolerance range in which motor function is maintained by the individual. Ectotherms, such as bees and other pollinators, depend on, let's go for it a little bit. Anyway, so CT max and thermal tolerance range are the most important, and as well as a multitude of other factors. So we talked about different ways in which bees manage their temperature, but one less explored way is the way that bees can manage their temperature through microhabitat selection. Temperature-driven foraging habitat selection is done by many ectotherms, and many ectotherms are known to effectively maintain their thermal optimum throughout their environment by doing this. This process is known as the Bogert effect, which is a term coined by Cowles and Bogert in 1944. For bees, their microhabitat selection is within their floral assemblages. Bees are known to use temperature difference in flowers to guide them to nectar, but less is known about their selection foraging area based off of the temperature of the area in total. Well, they're able to do this kind of selection because there's a lot of temporal variation within meadows and up to 5 degrees temperature variation with individual flowers. This variation in temperature has to do with floral traits such as structure, reflectivity, height, color, and so much more. Through this variation, bees have the opportunity to choose the area that best supports their thermal optimum while foraging. Because bees are ectotherms, the Bogart effect should explain how bees respond to temperature in a warming environment, with bees only selecting flowers that are a temperature that is within their thermal range. This information is extremely pertinent because a bee's floral preference indicates what flowers are and are not likely to be pollinated in a changing climate. So with all of this background information in mind, I developed the following aim and research questions. My research aim is to understand how temperature influences pollinator floral selection and how future warming will impact pollination. And my 3 research questions are, does temperature influence pollinator community floral preference in urban green spaces? What bee traits such as sociality and body size show resilience to urban warming? And do individual bees exhibit floral selection constancy based off of temperature? So based off the current scope of the available literature, I hypothesize that the higher floral temperature will be negatively correlated to pollinator abundance and diversity. Solitary bees will avoid heated flowers more often. Small bees will be more sensitive to temperature increases. Track bees will show constancy in flower temperature selection. And in the summer, when ambient temperatures are the hottest, hotter flowers will be visited significantly less often. So to test this question and hypotheses, I created an in situ heating experiment that emulated the warming that will occur in the coming century based off a scenario published by the IPCC. This experiment was set up at 6 sites with 2 different plots per site, plot A and plot B. Each plot had 4 plants, all of which were individually labeled. These plants were assigned to the plots based off the number of blooms and heights so that the sites all have relatively similar heights and number of blooms. Each collection day, I would rotate a heating treatment with an infrared battery attached to an infrared heater attached to a portable battery. The heater was set out at a 45-degree angle and warmed the plants for 10 minutes prior to collection and then 20 minutes while the collection period happened. The heater was rotated each collection day to reduce any impact of the heater on flower development. Each plant got an ambient temperature measurement apparatus, a setup I'll explain in a moment. And once everything was set up and in the correct place, a 20-minute collection period would begin. I replicated this setup at 6 different sites on the UTSC property. I had 2 sites on campus itself, 2 sites at the UTSC Valley, and 2 at the UTSC farm. All the sites represented 3 different kinds of urban space management, including an urban farm, a high traffic urban space, and a highly managed recreational green space. When visiting the sites, I would collect data in 3 different categories: temperature, plant characteristics, and insect visitation. For temperature, each plant had an ambient and surface temperature collection. So for surface collection, we used a FLIR thermal camera to collect surface temperature information after the plant had been heated for 10 minutes, which produced images like the one at the top there. Once these images were downloaded, we were able to use our amazing undergrad to draw polygons around each individual flower to get the average, maximum, and minimum of each floral display. Ambient temperature was captured using Credo sensors. The Credo sensors were placed in a paper cup to limit the impact of direct sunlight as well as wind. And then they were attached to a plastic pole at the average height of the plants. These devices would measure ambient temperature and humidity at 1-minute intervals. Additionally, we collected information from local weather stations, including temperature, wind, and cloud cover. When looking at our plants, we would simply count the number of blooms each collection day, but we would also take nectar measurements using microcapillary tubes. We took measurements from 3 flowers from each plant and averaged that value to find the average nectar per plant. Once this information was collected, we started a 20-minute collection period. We would collect all the individuals that visited the flowers, and if it was one of the bees that we had selected to be painted, one that was easily identifiable, we would pause the time and paint it using the apparatus shown in the video, so we could track them over the whole field season. Each bee that we collected, we did that through an insect vacuum, And we recorded what plant they went on, what time they did that, what site we were at, as well as a multitude of other pieces of information. And all this information helped correlate the insect visitation to the proper temperature measurements later. I repeated this experiment with 3 different plants representing spring, summer, and late summer. I used Penstemon digitalis in the spring, Asclepias incarnata in the summer, and Lobelia syphilitica in the late summer. These plants were chosen because they are all pollinator dependent, and they have very different pollinator communities. They also have very different morphologies, as you can see here. And additionally, these plants were able to remain in pots over the course of the experiment, which was a necessity as we worked in high traffic urban environments. If you would like more information about the setup of this experiment, I was fortunate enough to have a documentary made about my work available on YouTube. It was created by Mahora Tai from her time in the field with the team. Additionally, one thing to note is that all of the lovely photos and GIFs in this presentation were created by Mahora as well. So with the experiment, we collected 448 pollinators. We observed 617 flower visits, including 35 genera and 15 genera of bees, and we painted 96 bees. To begin the analysis, I wanted to investigate the effectiveness of the experimental warning treatment. I did this using t-tests at different temperature measurements to compare the heated and controlled plots. The difference between the control and heated treatments was significant at all levels, showing that the treatment was successful in warming the heated plants. The measurement that was most important to this investigation is the surface temperature of the flower taken with the thermal camera, shown all the way to the left of the screen. This is the most important because that's what bees perceive when they're making foraging decisions, and that's what the infrared heater most effectively warmed. Because of this consistent, significant difference between the heated and controlled plots, the analysis was able to use treatment as a large factor in the rest of the analyses. So to begin, I looked at how floral temperature influenced pollinator community floral preference, and I chose a multi-pronged method using a linear mixed model and a redundancy analyses. I used a linear mixed model to investigate the impact of floral treatment on abundance of all pollinators, including bees, flies, and wasps. I used site ID and plant species as random effects, and the treatment and total number of blooms per plots as fixed effects. This found that the treatment had no significant effect on the pollinators when you looked at all 3 experiments with all 3 plants together, as well as when we looked at each of the plants individually. I used an RDA to look at how Shannon diversity was impacted by treatment, and similarly, there was no impact on the diversity by the treatment. These results show that the urban pollinators that we collected showed more resilience to temperature changes than anticipated. The only aspect of the analyses that showed significance was the role of number of blooms on the abundance of pollinators, showing that the bloom number positively impact abundance. While there was not a consistent significant trend for all pollinators in their selection of floral treatment, there was a significant difference between taxa. This difference between groups was found through an ANOVA, which was then illustrated through a Tukey model. These tests looked at the different taxonomic groups and the average floral temperature selection from each taxonomic group. You can see in the Tukey that there are significant differences between the flies in both wasps and bees. Differences in foraging methods and life histories could lead to these differences in surface temperature preference. For example, the similarity between wasps and bees is most likely due to their similarity in life history as well as their foraging needs, which flies do not share. Flies are not central place foragers as wasps and bees are, and this could also lead to those differences. This result shows that diversity in temperature within urban spaces is important so that all pollinators can have their thermal needs met. I then took a more focused look to look at just the bees that were collected. Using the same approach as I did with all of the pollinators, I looked into the abundance and diversity and how it was impacted by experimental treatment, again using a linear mixed model and RDA respectively. When looking at the whole season, the treatment did not have a significant impact on the abundance or diversity of bees. But when I looked at each plant individually, I found that in the summer when temperatures were the hottest and I was working with milkweed, the heat treatment caused a significant decline in abundance of bee visitation, unlike in the spring and summer. This change in behavior by the bees is what I predicted through my hypotheses, and it shows that when bees are exposed to the highest temperature in most streaming heat events, they're more at risk for further warming. So after all the bees were collected and identified by Professor McIver, we were able to group them into appropriate sizes from extra large to small based on genera. So for example, carpenter bees were extra large and Sarrentina were small. Using an ANOVA followed by a Tukey model, we found that the large body bees visited significantly hotter flowers than did extra large or small size bees, with no difference for medium sized bees. More work is needed to be done once the bees come back from being ID'd to species. At that time, I'll measure integular space and redo my analysis so that I can use that continuous variable to look at the role of size and temperature selection more. In this thesis, I focused specifically on flowering plants, which already filter pollinators based on traits such as size. So studies that investigate broader patterns across flowering species and scale could show different results. Additionally, I looked into sociality. So once all the bees were collected, we assigned them social or non-social. And in order to investigate the impact of the experimental heating treatment, I used a chi-squared test to see if the social and non-social bees avoided the heating treatment. This test did not show any significant relationship between the sociality and treatment selection. This result is actually not consistent with recent studies, and this could be because urban bees do not have the same variation in temperature resilience when compared to their rural counterparts, which are typically the subject of the studies. More research is needed to see how differences between rural and urban bees and their climate sensitivity. We then went even further and we looked at specific bees. So we looked at individual bees by conducting a mark recapture experiment through common bee visitors during the field season. So this included Sarrentina, honeybees, bumblebees, leafcutter bees, and carpenter bees. We captured and painted 98 bees, and we had a 28% return rate. Only about half of the return visitors showed constancy in their floral treatment selection, and without further statistical analysis, we reject the hypotheses that bees show constancy in their selection. The lack of constancy might show that their floral choice is more strongly driven by different cues, such as nectar concentration, nectar volume, humidity, scent, and a multitude of different things. This activity helped build capacity in the lab, and I hope that more of these methods are used in the future by incoming lab members. With all the findings, we're able to now look back at the hypotheses. So we did not find that higher floral temperature was negatively correlated with pollinator abundance or diversity. Solitary bees did not avoid heated flowers more often. Small bees were found to be more sensitive to extreme temperature. The tracked bees that we tracked did not show constancy in their floral treatment selection. But we did find that in the summer, when ambient temperatures are the hottest, hotter flowers were visited significantly less often. This is a trend that was seen in bees, not all of pollinators combined. So in addition to the original tracks of questioning, there were some additional points that were found to be very important, including the role of high number of blooms on pollinator abundance. So when I was doing the linear mixed models and RDAs to investigate the impact of experimental treatment and the total number of blooms, the total number of blooms was used as a fixed effect, and it found that the number of blooms positively impacted the abundance and diversity of pollinators. The impact of high blooms was even more significant with bees, because bees are completely reliant on flowers, and their adherence to the optimal foraging theory makes them more likely to forage in large groups of flowers, because there's less of a chance of visiting a flower with no nectar. This finding illustrates the importance of high density blooms in a changing climate. This necessity would be met by having more heat robust species in urban spaces and taking greater care of prolifically blooming species so that they continue to support urban pollinators. Through the research for the experiment, another point was made clear to me. Air temperature measurements from weather stations, ambient air temperature, and surface temperature from study organisms are all methods that are used to measure plant temperature, but rarely do studies justify their choice of temperature measurements. This limits the use of these studies to support further work. And additionally, the measurement of microclimatic temperatures is less frequently investigated, especially in the field, making measurements like mine extremely valuable to coming researchers. Temperature is often viewed as one of the most simple types of abiotic environmental data to obtain. However, the complexity of environmental factors influencing the temperature, the variety of measurement approaches, and a prior knowledge of when and how temperature impacts a particular species or interaction needs development. Temperature studies need to be extremely context dependent, and so more work is needed to understand temperature as a driver of local environmental change, especially in an area of changing climate and urban heat island effects. So thank you to everyone who has supported me in the last 16 months and helped me wrap up this master's that I just presented. Thank you to the Bugs Lab and Professor McIver for all of their knowledge and help throughout the entirety of this. Additionally, thank you to my supervisory committee, as well as the staff at the UTSC Farm and Native Plants at Claremont, who really brought this project to life and welcomed it with open arms. And additionally, thank you to my parents, my friends, my partner, who have all supported me and told pretty much everyone about my painted bees over the last 16 months. So thank you all.
Great. Thank you, Charlotte. Usually at this time, just before we take a break and move into the smaller group of us to talk about questions, we can open it up to the entire audience if there's a question or two to Charlotte. Give it 5 more seconds.
It's pretty early.
That's well, I'm sure there'll be lots of great questions in the next session. So with that, thank you, everyone. Yeah, feel free to throw congratulations in the chat or what have you as you leave the room, and we'll just ask the committee to stay here.
Go ahead and stop recording now, Charlotte.
Yep.